Okay, thank you so much. Welcome, everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today, and I would also like to pay my respects to elders past, present, and emerging. Well, good afternoon, and welcome to the March FightMND Scientific Seminar. My name is Dr. Sarah Bennett, and I'm the Senior Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of the month. And last month in February, we heard from Professor David Berlowitz and Dr. Anthony Stell, who are working on a collaborative project using AI to optimize non-invasive ventilation for use in MND patients. If you're interested in this presentation and weren't able to join us live, a recording is now available on our website for you to catch up at a convenient time. Following the talk today, we are leaving plenty of time for discussion and questions. If you would like to ask anything to our presenters, please use the reaction button at the top of the screen to raise your hand, and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and read out questions on your behalf. Please note that the chat function is only enabled if you're using the app-based version of Teams and unfortunately does not work in the web-based version. So please ensure you introduce yourself and where you're from before asking your questions so we can all get to know each other. So now to our presentation for today. We are fortunate enough to have two speakers for today, joining us to discuss the capabilities at Phenomics Australia and then demonstrate how this research infrastructure can be applied to the M&D research sector. Professor Michael Dobbie is the founder and director of Phenomics Australia, beginning his work there in 2007 before stepping into the CEO role in 2013. His wealth of experience in the research sector spanned over 20 years prior to his Phenomics Australia stint, including postdoctoral positions in the UK. He is joined today by guest Associate Professor Shyuan Ngo, who is a group leader at the University of Queensland and director of the UQ Centre for M&D Research. She has a research program with strong focus on stem cell-derived neurons and organoids and joins us to discuss a vision for a national M&D stem cell research in partnership with Phenomics Australia. So thank you so much, Michael and Shu, for joining us today, and I'm really looking forward to your talk. Over to you.
Thank you, and good morning, good afternoon, everyone. Just start to share my screen. Is that visible for everyone?
Yeah, all good.
Loud and clear. Thank you for the invitation. I come to you today from the lands of the Ngunnawal people in Canberra, which Canberra comes from the Indigenous word for Canberra, Indigenous word Canberra meaning meeting place. So very pleased to meet with you all across the lands and the lands in which you are sitting today. And what I'd like to do today is also to talk to you about not only the research infrastructure at national scale, which is available through the organisation that I represent, which is Phenomics Australia, but across the whole of the National Research Infrastructure Program. And I'm very pleased today to be sharing the stage with Shu, who will be telling you, I think, about an exciting opportunity to expand the infrastructure to support NMD research. So what we've done today is to first of all, give you an opportunity to learn about what research infrastructure currently exists. Many of you, I know, have already used this infrastructure, but of course there are opportunities for many others to use that which already exists. And it has been in existence for nearly 20 years. And in fact, the program, it's the Commonwealth program called NCRIS, which actually stands for the National Collaborative Research Infrastructure Strategy, has been in existence since 2007. So we're now in our 20th year. And it's really this investment program, this operational program has been running for the 2 decades, and every 5 years it runs off a strategy, a national strategy, which is currently in review. And so you may have been aware that through last year, the program, the strategy was being revised by the federal government. You may well have participated in the ongoing consultations on that, and there is still more consultations to take place through this year. So certainly we'll keep you abreast of those opportunities, because I think this research community should have the opportunity to ensure that the roadmap, the strategy for provision of national research infrastructure properly attends to your needs. This is a broad program, and the purpose of the program is to develop and deliver the technologies and platforms, of course, but what it brings is the ability to deliver these at quality and at scale, beyond that which might ordinarily be available in your in the institutions in which you conduct your research. It looks to minimize duplication, but of course there is necessary duplication where we require scale or points of differentiation between the kinds of infrastructure, the resources that we develop and offer. But really, what I want to do today is to focus on access, because this infrastructure, while it exists, is not necessarily as widely known as it can be. And really just to present to you some of those points of discovery of what's available and how to access it. And importantly, it is a co-investment. So the co-investments come not only from the federal government, but from your state governments and from their host institutions, and also from the user fees. So many of the services that I'll describe just over the next few minutes are available on a fee-for-service basis, but they are subsidised. So at no point are you paying the full cost of the provision of that service because of these co-investments from the various stakeholders. So NCRIS is the infrastructure program. It consists of about 26 to 28 individual projects, and these can be discovered through a relatively new entity called Research Infrastructure Connected. And I'll come back to this in my last slide to leave you with ways to connect into the infrastructure. Now, the infrastructure is across all areas of research, not just biomedical research and certainly not just neurodegenerative disease. So we have several clusters across the program, and one of those clusters is the health group. That consists of 10 of the 26 or 8 projects, and these include, along with Phenomics Australia, the Population Health Research Network, European Molecular Biology Laboratory Australia, Platforms Australia, Therapeutic Innovation Australia, the National Imaging Facility, the National Fabrication Facility, the Microscopy Australia, the Australian Research Data Commons, and the National Deuteration Facility. And I've got a couple of slides for each of these facilities, just to give you a flavour for what's available. And we operate together to provide access in a very coordinated manner. So you might be looking for something, but then and successfully find that, but where will your project go? And the idea is that through working with a cluster, we can make appropriate introductions, we can stage access to different capabilities over time, we can work together to help support grant applications, to support individual research. So we meet, as a cluster, we meet monthly as directors. So we're very, very switched on as to what your needs might be. And so always invite you to make contact, and I'll, through this next few minutes, be able to provide those points of contact. So we'll just run through each of the different projects or capabilities. The first being the Australian Research Data Commons. There's a QR code there for accessing, and really they provide a lot of the policy work, but also specific resources to manage the various data sets, of which I know some of you are already using, such as through Research Data Australia. The second project or set of capabilities is the Population Health Research Network, which is focused on enabling linkage of data. So mainly this is clinical data, but increasing genomic data, cohort data, to ensure that you're able to access that data regardless of the jurisdiction in which it is held. The Australian National Fabrication Facility is probably right at the other end of your research, discovery and development and translation pipeline. ANF consists of 8 nodes, all around Australia. They have high quality, unique capabilities that enable specific fabrication in very clean rooms and at nano and micro scales. Again, more information can be found at their website, anff.org.au. European Molecular Biology Australia is largely a project that allows access to molecular biology capabilities, not only across Australia, but into, of course, the MBL's facilities across Europe. We are working with EMBL Australia to improve training and access to those facilities and international exchange for skills development between Europe and Australia. And I'm just featuring one recent recruit, William Roman, who's come to Monash University recently. And in particular, he has an interest in developing new technologies and pushing the boundaries of research in degenerative diseases, including motor neuron disease. So developing new cutting-edge technologies in cell-based modelling. And in fact is the recipient of a recent Pipeline Accelerator grant to look for new therapeutic targets for muscle atrophy. And again, I'll come to that voucher scheme, that Pipeline Accelerator grant scheme, which is open now. To my own space is Phenomics Australia. We consist of 90 technical experts, 40 research services across 18 locations, with 12 partners, half of them medical research institutes, the other half universities. And our areas of expertise are in engineering, customized engineering of model systems. So these are both animal and cell-based model systems. We also have high throughput screening technologies, pathology and phenotyping capabilities, as well as biobanking, which is one of the areas that we're focusing on at the moment, is to develop national biobanking capabilities and improving coordination. Again, you'll find more information at our website, but just to give you a feel for some of the model systems that are available. For example, we have specialization in customized bioengineered mice and rats. And also in more recently, in the last couple of years, we're now running the Australian Drosophila production facility, using, of course, all of the latest targeting capabilities and technologies. In our in vitro space, we have, of course, the full range of model systems. These are all currently under development, as you know, and our current focus is on standardization, improving reproducibility. And in fact, we will soon be launching a new stem cell registry. So that's one to watch out for. We do have high throughput screening capabilities, so we can take a lot of these cell models through to high throughut formats with very sophisticated imaging and assay development through a number of our locations. And we also have a histopathology, organ pathology, digitization service as well. And our footprint looks something like this. So this is where you can see that about half of our facilities are located in Melbourne, but they are also spread around the country. Of course, these slides will be available for share. There is a recording, so I won't dwell on these locations. I now turn to Bioplatforms Australia, which has 4 core platforms across genomics, proteomics, metabolomics, and most recently synthetic biology, all underpinned by a bioinformatics capability, which is operating as the Australian Biocommons, providing analytics. A little bit more detail, their footprint looks like this, with capabilities again across all of the major states, all of the major mainland states, and most recently have moved into synthetic biology capabilities. But what I'd like to do is to focus on the location in Macquarie University, which is a newly funded, including using with benefiting from support from FightMND, to establish a new national neuroproteomics facility, which is now online, open for business, there, and is working in collaboration with the Australian proteome analysis facility, also on the Macquarie University campus in Sydney. So I encourage you to look at those capabilities, which are now recently have become online through the Motor Neuron Disease Research Centre there. Turning to imaging, so this covers both human imaging and preclinical or small animal, large animal imaging, in terms of developing new pharmaceuticals and theranostics. There are radiopharmaceutical developments and capabilities, all serviced and supported by these various technologies. Again, these are Australia-wide, so this is the footprint for the National Imaging Facility. I'd just like to highlight a couple of projects which are MND related, which have recently been supported by the National Imaging Facility in particular, such as those that are using MRI to track brain changes in the brain during progression of neurodegenerative disease, such as MND. There's also other research which is being used to develop to detect the loss of motor neurons from the earlier stages of the disease, even before the clinical onset of symptoms. Microscopy Australia, again, is a national network of microscopes from all sorts of imaging and microscopes, and has up to over 250 instruments available across their network of 9 partners. Again, more information is available at their website. For those who are also interested in other radionucleotides, there's the National Deuteration Facility, which is part of the NCRIS network. This is useful for developing, again, new therapies. They have radio tracer and imaging capabilities. These are using some of the latest radio tracers, again, with brain-related biomarkers. And so I encourage you to think, if you're working in the clinical space, or maybe even the small animal imaging space, to think about some of these novel tracers which are available through the National Deuteration Facility. One of our other close partners is Therapeutic Innovation Australia, which offers capabilities across development of cell and gene therapies, identification and development of small molecule therapeutics, as well as biologics and vaccines, and most recently, of course, RNA and RNA-related products. Again, they have a broad national footprint covering all of these capabilities. Interestingly, they also have a technical feasibility assessment, so there's opportunities to have your ideas, concepts assessed through a consultancy option that is provided by Therapeutic Innovation Australia. One of our other colleagues in the space, so this is not NCRIS, is the Australasian Biospecimen Network Association. They represent a large number of Australia's biobanks, so effectively a network of biobanks. And very recently, they have relaunched an upgraded version of their specimen locator. So again, specimens that you might need for in considering how you might design an experiment or a set of experiments might already exist. And so I encourage you to go to the ABNA website and search for specimens that might be available through that new locator. So this brings me to an opportunity for support for accessing many of these capabilities, which is open now. I'm closing in a couple of weeks' time on the 17th of April. This is the Pipeline Accelerator Voucher Scheme. This has been running now for about 3 or 4 years. We have been partners to this in the last several years. And in addition, there are vouchers available to not only access therapeutic innovations capabilities and our capabilities, but also those more recently for Bioplatforms Australia and the National Duration Facility. So either go to this URL or use the QR codes to go to either TIA or Phenomics' website, where you'll find the guidelines, application forms, etc. So these is open every 6 months, and practically what this provides is up to a 50% reduction in the access fee to access many of those capabilities that I have just highlighted, up to a maximum of $50,000. So, for example, if you are looking to commission the production of a mouse, that might ordinarily cost you $10,000 or $20,000, that would reduce the access fee, if you're successful through an award of a voucher, to half of that access fee. Or, for example, if you're conducting a drug screen or a drug target screen using one of our high throughput capabilities, which might be a $100,000 scheme, or maybe a $150,000 project, then there are access vouchers to reduce the cost by up to $50,000. And then finally, I'll leave it here. So this is where I started. So there's Research Infrastructure Connected, which is now a new way to discover much of the research infrastructure, the national research infrastructure, which is available. So even if there's not something in your own facility, institution, there are ways to discover what might be available and to access it wherever that is in Australia. So I'll pause there. I think that's a good time. I'd actually like to leave plenty of time for discussion. I'm probably take questions after Shu has spoken, because I think there's other aspects as to not only being able to better access and know what's currently available, but to get us thinking and working together about how we might collectively as a community identify what's missing and what else we should seek additional funding and support for to build even better resources for MND research. Thank you. I'll stop share.
Mute. Can I just confirm that people can see the slides?
Yeah, all good.
Okay, cool. Thank you. Thank you, Michael, for that. That was fantastic. And I'm just going to go through today a vision that we have that I've been talking to Michael about and several other MND researchers and non-MND researchers across the country about what we might like to achieve in terms of a stem cell resource. I'm Shu, representing UQ Centre for MND Research, but I think MND research as a whole in Australia at the moment. So one thing that we know that is happening globally is that there's change on the horizon. And in April in 2025 last year, the FDA announced that they were planning to phase out animal testing, and they were wanting to focus more on human-derived models to be able to facilitate monoclonal antibody development and drug development for several diseases. This was followed soon after by the European Commission, who have also developed a roadmap to phase out animal testing in research with a preference towards moving towards human-based models. And a good example of this is that recently in Utrecht, there was a 250 million euro investment into the Brain Research Centre, specifically to develop human cell-derived models for disease modelling and drug development for several brain-related diseases. So where does Australia really stand in terms of this space of transitioning away from animal models to human cells? Well, over a year ago, there was this piece in The Guardian saying that, the world is making this change, and will Australia get left behind? And in fact, over the last year, there hasn't really been much progress made in terms of where Australia sits in their consideration of whether or not we transition away from the use of animal models and more towards these cell models, particularly when we are trying to address the many human-related diseases that face our aging population. So is there an unmet need for MND research in Australia? Well, what we do know is that across Australia, what a lot of researchers do is they work closely with people who are living with the disease, but they also use several laboratory-based models to address research questions. That is not only to try to discover the mechanisms that are driving the disease, but also for developing therapeutics. And this is not to say that I'm all for moving away from animal models towards cell models, because what we understand, and I think what FightMND is a strong proponent of, is that for us to be able to tackle a disease as complex as MND, we require a combination of multiple models to validate our findings. And the research questions that we address really should arise in most part with people who are living with the disease at the core of that research design and research question development. And then we can then go into the lab and use a range of models, whether they be blood cells that are isolated from the people living with MND themselves, stem cell derived cultures, including 2D cultures and organoids and muscle cultures, as well as mouse models and post-mortem tissue. And with this large array of models or samples that are available to MND researchers in Australia, what I want to focus on today is the stem cell models. Someone's using a mouse for me. The stem cell models. So as a first step, what I did was I sort of took a step back and looked at and tried to think about who in Australia are custodians of stem cells that are used for MND research. And some of you may know the faces here. We've got my lovely mugshot up here in the top for Queensland. We've got Professor Lizanne Ooi down at Wollongong in New South Wales. Kathy Blizzard has some stem cells down in Tasmania, although hers are not derived specifically from people living with MND in Australia. Brad Turner and Chris Bai collectively are custodians of the largest collection of MND stem cells in Australia, with over 150 stem cell lines. And then over in Western Australia, we don't forget the West Coast, we've got Professor Anthony Akari, Dr. Lauren Flynn, and newly minted Dr. Lian Zhang, who are managing the stem cells who are donated by research participants over on the West Coast. But the question really is, are these all of the stem cells that are available for MND research in Australia, or are we missing something? Are we not capturing all stem cells that have been generated from people living with MND who have specifically donated for us to advance our research discoveries and to find treatments for this disease? So let's take a step back and think about why stem cells are so important. And I am crediting these slides to my close colleague, Christine Wells from the University of Melbourne. Now, pluripotent stem cells, of course, are cells that can be made from anyone and any somatic cell, so any adult cell. We can make stem cells from skin cells, we can make them from epithelial cells that are shed through the urine, or we can make them from PBMCs. And because these cells are pluripotent, they're essentially self-replicating, and we can generate millions and millions and millions of these cells, which means we can bank them and we can share them with researchers as long as ethics approvals allow us to do so. Now, the power of these stem cells is that they can be grown into most tissues of the human body, and they can reveal a lot about the disease state of the original donor. This means that it opens up a lot of opportunities for driving precision medicine and also the emerging use for tissue transplantation. And I'm not sure if you know, but there's been a recent publication in Nature describing the successful transplantation of iPSCs into the brains of Parkinson's disease patients with minimal side effects and adverse events. And so, times are changing, and these iPS-derived models are very, very powerful models, not only for studying disease and drug development, but hopefully in the not so near future for tissue transplantation. But having said that, we have a challenge. Yes, we know that these cells can be used for disease modeling. We can use them for drug screening and regenerative medicine and cell therapies. But the problem is, and I don't know whether this is relevant for other fields, but certainly for M&D, is sometimes we don't know which cell line we should be using. We don't know where to find a protocol that is easily reproducible and that works. And we don't know whether or not, once we have this stem cell, whether we can make a cell type of interest, and for M&D that would, for example, be a motor neuron that exhibits the disease-related phenotypes that we want. And so there are a lot of questions when it comes to IPS, and sometimes it can cause some researchers to shy away from their use because the use of stem cells is expensive, it's time intensive, it's resource intensive, and it requires, in some cases, really unique and very good aseptic technique and expertise within the cell culture arena to be able to generate cells that we can use for disease modeling. Now, if people were to try to want to use a stem cell for M&D research, where would they even find the information of where they can source a stem cell from? If they weren't working with either myself or Lizanne, Brad, Lauren, Anthony, and Kathy, where would they go to? And people who are new to the field of M&D research, where would they be able to find these cells? And so we proposed a solution, Christine and I, with also researchers down at Peter Mac and another one at UQ. We proposed a solution called the Pheno Bridge. And this is currently under assessment as a grant, and hopefully we will get it, but we can't always be sure with grants these days. But the Pheno Bridge is a way to link several capabilities that will allow us to drive stem cell discoveries and cures. And one of the first infrastructures and capabilities that really sets the foundation for Pheno Bridge is the Australian Stem Cell Registry. Now, the purpose of the Australian Stem Cell Registry is to map all of the pluripotent stem cell lines that have been generated from Australian donors and that are used in Australian laboratories. So this means that once a stem cell is made from a donor, it can enter the registry, be given a unique identifier, and then tracked throughout all use, whether that's through manuscripts or through research labs or through grant applications, that we can see where that line is going and what it is contributing to, because it has that one unique identifier. Some people may have tried to look for stem cells before, and they may have found that a stem cell is named classically MND1 in someone's lab, but when it's published and entered into a registry, it's given a completely different identifier. And so it's actually quite tricky to track and link data for some of the stem cells that have already been generated in Australia. The second platform is Stemphomatics. And what Stemphomatics is, it is a web-based tool that allows individuals to visually explore data sets, particularly omics data sets that they've generated, in a quick and easy way. And what it does allow also is that it allows researchers themselves to benchmark their own data sets against more than 400 manually created high-quality public data sets that are made available through researchers globally. And this means that once you generate an omics data set from your stem cell lines, for example, you can go into this platform and see how your data stacks up against data that's been generated through other stem cell labs globally. The other data or infrastructure is the CM portal. So the CM portal has been developed by Professor Nathan Palpant at the University of Queensland. And initially, it has been specific for the generation of cardiac-derived cells. But what we're hoping to do is to expand the CM portal to generate neural cell types as well. And so what the CM portal will serve to do is it will be a resource of validated protocols that will generate specific phenotypes. We know that there are several protocols that float around the lab for generating motor neurons, for example. They can be very difficult to replicate across labs, even within labs, and to generate the same phenotype is even more tricky. And in actual fact, most stem cells that are used for M&D research are likely used so that they can generate 3 key phenotypes at this point in time, I think. Neurite outgrowth, changes in cell survival, and TDP43 mislocalization. But these 3 phenotypes may not necessarily be the most ideal for answering research questions for all researchers across Australia. And so for CM Portal, what we want to do is collate all of the validated protocols that will give a diverse range of phenotypes for ourselves so that people can go into this portal, find the right protocol for them, and start to generate their own cell types from stem cells that they have identified through the Australian Stem Cell Registry. And they can link everything that they do in their lab with any omics information that is already available for these cells. Now, a key aspect of PhenoBridge is this dynamic consent. And the dynamic consent platform, briefly, is a way that we want to engage people living with M&D at every step of the process in the use of their stem cells. So the platform allows us as researchers to provide more appropriate, granular, and flexible consent options for people who have donated their cells. And it means that people who have donated cells can also access study information. They can see what is being done with their donation, and they can see which labs those donations are going to and what papers have actually been generated from their samples. It allows people with M&D or people who have donated these cells to have increased scientific and medical literacy because they have very close face-to-face contact with the research teams, but also access to those publications. And there is constant two-way communication between participants and researchers, where researchers can constantly feed back information to the participants of how their donations are contributing to research progress. And overall, this really is to build trust in the process so that when a donor provides a sample, they know that it's being used for something, that it's not sitting in liquid nitrogen in one of the many universities across the country, or it's not sitting in a freezer somewhere. Now, for research organizations, the dynamic consent is really important because it allows us to have electronic consent records. And it allows us to retain individuals in longitudinal studies because of that two-way communication between the participants and the researchers. There's clear data sharing frameworks of health information, and it really helps us address ethical, legal, and social issues that are relevant to stem cell studies, but also the genomics data that comes from any stem cell and derivative that might be generated as part of research studies. So in terms of the broader picture of how it all fits together, what we want to see is a dynamic consent process where people who are donating samples are constantly involved in the research progress, not only in reviewing the research progress, but potentially also contributing to other research projects. The dynamic consent platform will allow people to go into the stem cell registry to see what additional projects might be available, which they can then consent to have their lines shared with. Now, with the stem cell registry, we will have a digital ID for that source donation, and that digital ID will follow any derivative that is generated from that IPS line, whether that be a derived tissue, organoid, or IPS. Feeding into this stem cell registry, we will have private information that is locked behind many security processes that Christine is very familiar with because she has done this before for Stemfematics, which can be linked to any of the cells that are generated should researchers want to link that information, and as long as the sharing of information is approved ethically. There'll be public information that can be shared. This includes publications that are already generated and public data sets that are available for these stem cell lines, but also related but distributed data. So any other published data, whether that be in-house phenotype data that researchers would like to share, will feed into the stem cell registry and essentially give us a really in-depth map of what each stem cell and its derivatives will look like. So how do we plan to do this for MND in Australia? So we've started to canvass individuals across the country who might be interested in being involved in such initiative. You will see here that we've got the usual MND players. I'm hoping that there may be other MND researchers in Australia who do have stem cell lines and who want to contribute to this initiative. We have researchers who are not MND researchers, but have a lot of expertise in disease modeling in other neurodegenerative diseases, but also Christine, who is an absolute powerhouse in developing platforms that are designed really to facilitate research and the sharing and integration of data. So the vision for the Australian MND stem cell repository is really to have a centralized location where all of the stem cell lines that are available for MND research are located. These lines can either be clonal in nature or non-clonal. Those that are non-clonal will be made clonal through StemCore or NeuroStem based at the Florey. StemCore is an NCRIS facility based at the AIBN where I am. And they can essentially make these clonal cell lines, confirm that they're pluripotent, confirm that these cells will generate the 3 germ layers that they're meant to, and then these can be karyotyped, have very high quality QC before they are then banked either at StemCore or at Cell Bank Australia, which is also an NCRIS facility. We want to, of course, partner with Phenomics Australia, and you can see the breadth of expertise and capabilities that Phenomics Australia brings to the table through what Michael has presented just earlier today. Now, building on top of these huge capabilities, NCRIS capabilities for making these lines and for storing these lines, we will bring in the Australian Stem Cell Registry. So all of these lines will be registered so that we can search them within Australia for any researcher can search them. We will link them, we will link in Stemfematics, so any of the omics information that is generated from these cell lines will be linked in so that this can be used to inform research outcomes from studies. So for example, say that someone has generated motor neurons from 10 stem cell lines and they've done a drug screening, a pilot drug screening project, and they've noticed that some lines respond better than others. What they can do is then go into Stemfematics, pull any relevant omic information, which might further inform what they are seeing from their drug screening. We'll obviously have the dynamic consent platform because for us, it's really important to ensure that people living with MND maintain engagement with their donations. And then finally, we hope to partner with FightMND and MND Australia, but also link in current clinical data, whether that's cross-sectional or longitudinal, that is available through the SALSA consortium. So we can link clinical data to the stem cells that have been donated so that, again, research outcomes can be linked back to the clinical phenotypes of the donors at baseline and over time. And also the MindOz partnership, which is patient-reported data, and we can link in what patients are reporting about their own disease and their own condition and their own journey back to the stem cell donation as well. So that is the end of my presentation, and I'll stop sharing. But really what we're hoping to do today is to discuss or to garner your thoughts on what you think an effective Australian MND stem cell repository would look like, whether you would like to be involved in such an initiative, whether you think it's important. And this is what, sorry, it's open to questions and feedback and criticisms, although if you're going to criticize me, please be nice.
Yeah, thanks, Michael. Thanks, Shu. Really appreciate your discussions and presentations, Michael, over the overview of the organisations and facility under the NCRIS umbrella, and what they can provide for our research communities, and also Shu, an amazing presentation there in concept and design around the Pheno Bridge, the dynamic consent process. I'll just say it's open for discussion, so please raise hands digitally, and Sarah will let you ask your questions. But I'll go through the first general kind of question to Shu around the dynamic consent process. And are there other, I understand what we're trying to do here is get this to be MND, specific MND focused. Are there other disorders, neurodegenerative disorders, that you've learned from to come up with the consent process, or is this some new design that you and Christine have come up with?
Yeah, so the dynamic consent platform, maybe under embargo, maybe not, has been successfully funded, and so that will be implemented broadly for stem cell research in general. And so we want to capitalize on that funding through that MRFF to integrate that into this Australian MND stem cell repository, so that we are not, I guess, duplicating efforts for dynamic consent. And so it won't just be specific for MND, it will be for all stem cell donations in general. At this stage, we will be looking at cardiac diseases, cancer, and motor neuron disease as those sort of flagship diseases, but this can easily be transitioned into any other disease in Australia.
Yeah, thanks, Shu. Mary Louise, you've got a question, so Sarah will assist you in allowing your voice to be heard.
Can you hear me?
Oh, yeah.
Yes, your voice is heard, Mary Louise.
It's Mary Louise from Flinders. I'm just interested, Shu, that, you know, when we were about the phenotyping, you know, that when we were discussing globally or the problems, and one of the issues, I mean, I'm not in the stem cell world, but I know that that's been discussed globally about making sure that everybody has the same sort of protocols and things. Is that something you've considered? You probably have. I'm sure you have, but
yeah, so
you know, globally in that.
Yeah, so that is certainly something that we've considered, and over the discussions thus far, depending on which stream we go on with for the MRFF, we're thinking of applying for, you know, this critical research infrastructure, and there's two streams. There's a $3 million stream and a $7 million stream. I think irrespective of which stream we go for, there are three key things that we want to be able to achieve that I think will be achievable, and I think will be a big win for MND research as a whole. The first is to be able to collate all of the stem cell lines that are available for MND research in Australia, and to know that they exist and to have this central repository. The second is to confirm that all of the IPS that have been generated will differentiate into the three lineages, because that is the minimum that we were required to ensure that our IPS will do what they want to do. And the third, which speaks to your point, Mary Louise, is that we want to establish a minimum standard or number of phenotypes that our IPS can achieve before we will release them to researchers, because we don't want to give them a cell that won't do what they want it to do. This builds trust in the process. But in order to come to a decision on what those phenotypes are, we need to know what phenotypes are actually being assessed across Australia. And we need to come together as a community, not just the researchers themselves, but people living with MND in a workshop type of setting, to decide which are the phenotypes that we think are most important for looking at disease mechanisms and drug discovery. as long as we hit those minimum phenotypes and standards to start with, we can build on that over time, because obviously those initial minimum phenotypes aren't going to be entirely relevant for everyone. And we might want to look at other phenotypes, including, autophagy or lysosomal pathways, or, I don't know what else would come off the top of my head. I'll stop there.
Thank you.
Thanks, Marie-Louise. Thanks, Shu. Comment from Philip Camden. Hi, Phil. Hope you're going well. Saying that loves the dynamic consent process. And Shu, just a comment on, keeping Plex in the loop on that front. If you got further comments around that.
Yeah, Phil, we do want to keep people living with MND in the loop. And so I'm hoping that with the next discussions, when we Christine and I start to formulate what this dynamic consent will really look like for MND, that we will be engaging, because we have engaged with certain individuals previously, which I'm sure you're quite aware of, is to continue that engagement, because, we have an idea of what this dynamic consent platform will look like. But for people with MND, I think the dynamic consent will look quite different, potentially for someone who's got a longer living sort of disease. And so that's something that we need to make a bit more bespoke for MND.
Thanks, Shu. Michael, I'll bring you into the conversation on the Phenomics Australia front. And coming back to, from a funding perspective, the voucher system, in terms of, I can use an example of generating a stem cell line, but I'll go back to generating, with the discussion here, but generating a model of disease, for example. How does that work? How would that work in terms of, is there an application that comes through the voucher system and then they get assessed on merit, but even if successful, what happens to the model afterwards?
Yeah, thank you for the question. So part of our core business is about being able to engineer these lines as appropriate, whether they be patient-derived or otherwise. We have several nodes, service nodes, that provide variations in specialties, so we'll be able to make introductions to those nodes now. That you will be able to design what's needed. We would provide, or our node would provide a quote, and that then quote and the design would form part of the application into the voucher scheme. And the successful awardees, again, it's simply merit-based according to the guidelines, which are available clearly there, would be assessed, and then the awardees then would be able to proceed. The idea is that the project would start, I think, normally within about 6 months of the award of the grant. Effectively, it's a cash subsidy which goes straight to the service node to reduce the cost of access to that service.
Yeah, can I ask a question?
Yes, sure.
just for clarity. So let's say I want to apply for a voucher to make an organoid model. Would that be something that we would do in collaboration with Phenomics Australia and the organoid facility that's based at the AIBN?
Yes.
Yeah. Okay.
Yeah. So if it's one of our facilities, then it would be eligible for a reduction in the access fee, in the cost of production of your resource. And then what happens afterwards? Who owns it? It's up to the researcher really, as to at what point they would like to work with it. As an NCRIS capability, as an NCRIS provider, we have no need or intention to hold any of the IP. The IP remains with the commissioning researcher, probably actually with their affiliated institution that owns everything. But given that it was generated using public funds, whether they be research council funds, philanthropy, or funds coming through NCRIS, or cash contributions that come through the institution itself as a co-investment. These are public resources, and we really strongly encourage these resources to be available at an early stage. Ideally, I guess the IP would be secured by the researcher and their institution at the point of publication or some sort of notification about the data. But we would seek to at least capture the data, or not the data, but information, the metadata around that resource at an early stage, but not release that publicly. So we have we would have anything that would be banked, for example, through any of the banking resources would be banked at an early stage, even if it's not publicly discoverable at that stage until the IP has been secured by the institution.
Thanks, Michael. I was going to ask, probe further into governance barriers, especially with different institutions being involved with the project you were and program you were talking about, Shu. But there's a question that's come up from Kate. I'm not sure where Kate's from. Maybe Kate can put that in the chat as well. But Kate's an honours student, just starting in the MND space, and she's asking either of you to explain how the stem cell registry could help researchers, especially less experienced ones, choose the most appropriate stem cell line for their research.
Yeah, that's a great question. This is what we're hoping the Australian Stem Cell Registry will do. And this really comes from the integration of the stem cell registry itself, where each stem cell line is given a unique identifier. The Stemformatics platform, which allows you to benchmark your data, but the CM portal, which is essentially the infrastructure that we hope will hold all of the protocols that you need. And so ideally what you will do is you will come into the stem cell repository, you will search, you want you would like to find 5 stem cell lines that have been shown to generate motor neurons that have X phenotype. Then that would come up. Those filters for your search would then pull out the stem cell lines that are available in that repository, which you could then go and use, or that you could go and contact the custodian to then get approval to use. And then if the custodian agrees to and the ethics allows for that, or we go back into the dynamic consent process and can see that that is allowed, those cells will be dispersed to you from either StemCore or Cell Banks Australia. That's how we see it happening.
Very, very
Thank you so much for your answer.
I'll throw to Sarah to wrap up.
Yeah, thanks everyone. Thanks Michael and Shu, especially for taking the time to speak to us today. I hope you've inspired the Australian M&D research community what might be possible when nationally available research infrastructure is utilised and we work together. So if there's anything else you'd like to follow up with either Phenomics Australia or the stem cell resource, and they would be very happy to help you, to work with you to identify how they can help advance your research. So reach out them directly or to us at FightMND if you require any assistance. And obviously, yes, as Michael mentioned, that the voucher grants are open at the moment. So if you would like more information on those and would like to any help in understanding the application, they'd be very happy to speak to you. So thank you so much for joining us today for the March FightMND Scientific Seminar. I hope you've enjoyed today's presentation. We look forward to you joining us again next month, where we will have an exciting new seminar, and this will be on Tuesday the 28th of April at 12 p.m. Australian Eastern Standard Time. So thank you. Until next time, we'll see you then.